Hello, today's day one and we will understand the genetics world inside us. First of all, we are all different because we have different traits, and these traits are determined by the genes we have. For each gen, we have different gen variants, which are called alleles. There are two types of alleles, dominant or recessive. Each parent gives you one allele. For the dominant alleles, you only need one copy to express the genetic trait. For recessive alleles, you need two copies. Here are some examples of the dominant allele, which can be curly hair, dimples, or cleft chin. On another hand, an example of a recessive allele can be detached earlobes.
Let's have a look inside the cell. So inside the cell there's DNA, which is formed by complementary nucleotides. A is complementary to T and C is complementary to G. They carry the information to produce proteins. There's two types of proteins, the structure protein and enzyme protein. To extract the DNA, we can use water and detergent because they have similar molecular structures, so they combine together and are able to break through the cell membrane, which is made of hydrophilic and hydrophilic components.
From DNA to mRNA to protein. This process has two parts, the transcription and translation. The transcription happens in the nucleus of the cell. And when the DNA opens, it's a signal for the RNA polymers to make an mRNA copy of that part of the DNA. The translation is performed in the cytoplasm, and the mRNA comes out from the nucleus to the cytoplasm, where it goes through the ribosome and it is converted to amino acids. Finally, the amino acids are folded into a shape, thus forming a protein. Electrophoresis. There are 3 steps to make an electrophoresis. First, you cast the gel, then you load DNA sample, and then you run. In the picture, we show how the smaller DNA samples move faster than the bigger ones. Run means the step in where the DNA samples move. This happens because the DNAs that are negative attract to the positive when we put electricity, and that's why they move.
Epigenetics. The epigenome is on the top of the DNA. That's what epi means. It is a set of molecular switches that turn genes on and off. Acetyl groups turn on and methyl groups turn off. We have the same DNA in all the cells in our body. Not all the information is needed in every part of the body, so the molecular switches turn on 2 genes we need in specific parts of your body and in different times, for example, the caterpillar and butterfly. Factors in the environment can affect the turning on or off of the genes in the epigenome. For example, chemicals, exercise, stress, and diet.